start our look at reactions of carboxylic acids and their syntheses, I want to return to the reaction that I introduced this chapter with, which is the deprotonation of carboxylic acids. Strong bases, hydroxide bases for instance, can completely deprotonate carboxylic acids to produce carboxylate salts. So in the same way we make soap, if you use low molecular weight carboxylic acids, you'll simply produce the carboxylate salt from these by reaction with hydroxide salts. This really has two effects. Most importantly is the much greater water solubility that the carboxylate salts have. If you take acetic acid, for instance, like is shown here, deprotonated with sodium hydroxide, you form sodium acetate, which has very high solubility in water. The other thing that changes dramatically is the vapor pressure. So the vapor pressure of the carboxylic acids are such that they typically have very strong odors. Acetic acid, for instance, is the thing we associate with the smell of vinegar, can be very potent. The carboxylate salt, on the other hand, has virtually no vapor pressure, has almost no odor at all. This is particularly important for the slightly longer chain carboxylic acids, which really have some displeasing odors. Things like caproic acid and things like butyric acid really have foul smells, but simply neutralizing them produces something that has virtually no odor at all. We've seen ways of synthesizing carboxylic acids in the past. Just a quick review. Most of these are simple oxidation reactions using chromium oxidants. You need a strong oxidant in order to do this. So for instance, primary alcohols are typically cleanly oxidized to carboxylic acids using dichromate or chromic acid solution. Usually this is done under acidic conditions to create the chromic acid. Presumably the intermediate in the reaction is an aldehyde, and the aldehydes themselves can be oxidized. The other reaction that we learned is side chain oxidation of alkyl aromatics. What's required is that you have at least one benzylic hydrogen, and treating these again with chromium oxidants or manganese oxidants will produce a corresponding benzoic acid derivatives. There's one other way, which we haven't touched on yet, but it's something known as the iodoform reaction. This is a reaction of a methyl ketone. We have a ketone here that has one methyl group attached to it. Treat this with a mixture of iodide, elemental iodide, under basic conditions, and you get a periodation of the methyl group, and then ultimately a displacement that produces a carboxylate salt. This is a reaction we'll talk about later on in a subsequent chapter. So some other ways you can make carboxylic acids involve the use of Grignard reagents and reacting them with carbon dioxide. You might recall there was a post-lab question that asked about this. Actually, asked you to draw the mechanism of this, and most people get that right. It's a fairly simple mechanism. The carbon dioxide here can be either carbon dioxide gas, it's bubbled through a reaction mixture, or you can use solid carbon dioxide, better known as dry ice. You simply add it to a reaction mixture, and you get very clean reaction. The immediate product is the carboxylate, the anion, as the magnesium salt. And so to work these reactions up, you do have to add aqueous acid to protonate and isolate the actual acid. A little bit later on in the semester, we're going to talk about this reaction in more detail. This is hydrolysis of nitriles. We know how to make nitriles doing typically substitution reactions. So we make compounds of this sort, for instance, by using cyanide and just simply doing an SN2 reaction on an alkyl halide or a tosylate. Under vigorous conditions, strongly acidic, strongly basic, usually heat, you can hydrolyze nitriles into the corresponding carboxylic acids. This is in fact at the same oxidation state, so there's no redox chemistry going on here. It is a hydrolysis reaction. So if you think about this as an overall scheme, here's an example of that. Take a primary halide, treat it with cyanide under SN2 conditions, polar aprotic solvent, for instance. We get substitution to give the nitrile, and then an acidic workup. Acid hydrolysis allows us to convert that into a carboxylic acid. So that makes a very convenient way of synthesizing carboxylic acids. We'll talk about the mechanism of this process in a later chapter. So let's talk a little bit about reactions that carboxylic acids undergo. One of the more important reactions that they undergo is the conversion to acid chlorides, and this can be done cleanly with either of two reagents. One of those reagents is thionyl chloride, which we see here. The other is oxalochloride. They're more or less interchangeable. These are inexpensive, readily available reagents. They are very reactive. They have high vapor pressures. They react with water, for instance, so they have to be handled under special conditions. But typically you get very clean conversion into the corresponding acid chloride. Yields tend to be very high. There's a very strong driving force for these reactions to occur because the byproducts, and you can see that in the specific examples down here, the byproducts of these reactions are gases. And the release of those gases drives these reactions forward, essentially meaning the reactions are occurring under non-equilibrium conditions, going to completion. If you use thionyl chloride, the byproducts are sulfur dioxide and hydrogen chloride. And if you use oxalochloride, you actually get three byproducts: hydrogen chloride, carbon monoxide, and carbon dioxide as byproducts. So what else can occur with alcohols? Well, we actually have learned a couple reactions earlier on. One of those is reduction. Lithium aluminum hydride will reduce carboxylic acids. Importantly here, typically not sodium borhydride. It's a less aggressive reagent, so you need a stronger reducing agent. Lithium aluminum hydride is a stronger reducing agent than sodium borhydride. Typical conditions, under anhydrous conditions, you react the lithium aluminum hydride with the acid, and then an acidic workup allows you to isolate the primary alcohol. And notice here, you're always going to end up with a primary alcohol because you're reducing this carbon. It kind of by definition would be at the end of a chain, and so reducing that down to the alcohol, adding two hydrides to this carbon means we're going to end up always with a primary alcohol. Another reaction we learned is condensations of carboxylic acids. This is just a shorthand way of drawing carboxylic acid, COOH. This can react with alcohols under Fischer esterification conditions and produce esters. Now I introduced this reaction in the alcohol chapter. We were talking about condensation reactions of alcohols. It's time to learn what the mechanism of that reaction is. So what is the mechanism of a Fischer esterification? In 1938, a classic experiment was done that used oxygen-18 enriched alcohols and reacted those with carboxylic acids. So the notion here is that this oxygen in our alcohol, in this case methanol, was synthesized in a form where it contained the unnatural isotope of oxygen, oxygen-18, so you could trace that throughout the mechanism. The researchers, Roberts and Uri, discovered that the oxygen-18 was incorporated into the final product, in this position, the oxygen of the ester. It did not end up in the water produced from the reaction. So our mechanism has to account for that. So what is the mechanism of the Fischer esterification? So the reaction begins with the protonation of the carbonyl of the carboxylic acid. We use a strong acid to catalyze this reaction, so our proton donor would be protonated methanol. Protonation of the carbonyl gives a more electrophilic carbonyl, and methanol will do a nucleophilic addition to that carbonyl. This produces an intermediate of this sort, which is deprotonated by the methanol in the reaction to give an intermediate, a neutral intermediate, sometimes referred to as the tetrahedral intermediate because the carbon at the center now is tetrahedral. This is then protonated on one of the hydroxyl groups, which leads to a loss of water. The elimination of water essentially reproduces the carbonyl, albeit in a protonated form, and it's deprotonated to give the final ester. And you'll notice here that each of the steps is indicated as an equilibrium. And you'll recall when we talked about this reaction in the previous chapter, it is an equilibrium process. And so if you carry this out, for instance, just in a closed container, you get an equilibrium yield. But you can push the reaction by carrying out under non-equilibrium conditions by removing the water, for instance, from the reaction. Also, the process is completely reversible. So one can take an ester and hydrolyze the ester, reversing the entire process to yield an alcohol and a carboxylic acid. You should take it as a steady exercise to write the mechanism for the reverse of this process, the hydrolysis of an ester. We're going to visit that again in a later chapter. Esters can be made in rings. If you have a carboxylic acid and an alcohol in the same molecule, you can form rings. This can be done with small rings, and even medium to large size rings can give reasonably good yields of these cyclic esters. Historically, these cyclic esters are given the name lactones, but they really are just esters. So you can see in our first example here, we have carboxylic acid, and at the other end of the chain we have an alcohol. Just treating that with a catalytic amount of acid, we actually get, and these are equilibrium yields, we actually get an equilibrium yield here of 73% because the 5-membered ring is reasonably stable. Larger lactones are not as entropically favorable, and so you don't get a very good yield of them at equilibrium. But if you put in an azeotroping solvent like benzene and distill out some of that solvent, it will remove the water and thereby push the reaction past the equilibrium position, and you actually get very good yields. So you can see in this case our 10-membered lactone, we can get a 95% yield. There are lots of cyclic esters, lactones, found in nature. Vitamin C exists essentially as an equilibrium between these two structures. This is the open chain form of ascorbic acid, better known as vitamin C. This is the closed chain form of it. There are chiral centers in ascorbic acid. I've kind of ignored those right now. There are 2 chiral centers here in the structure. Under physiological conditions, it largely exists in the lactone. I've mentioned this molecule many times. It was the target of my graduate research, amphotericin B. It belongs to a class of antifungal antibiotics called the macrolide antibiotics. And the macrolide refers to macrocyclic lactones. It is a very long ring, and you'll notice right here, here is the ester linkage that makes this, in fact, a lactone. Our next reaction is a reaction that is unique to particular structures, and it is a decarboxylation of beta-keto acids. So not just a carboxylic acid, but in this case specifically a carboxylic acid. Here's our carboxylic acid structure, and I've drawn it this way for a reason. It lies at the end of a chain. The immediately adjacent carbon is known as the alpha carbon. This is our reference position, and then the first position adjacent to it is sometimes called the alpha position. And then this position is beta, and so on down the chain. If we have a carbonyl beta to a carboxylic acid, these sorts of compounds are thermally unstable, and typically thermally unstable even at very modest temperatures. Just warming slightly, or even at room temperature, many of these compounds will spontaneously undergo a decarboxylation reaction. The result is you form a methyl ketone. In this case, you can actually have substituents attached to the alpha position, and they're retained. But the byproduct of the reaction is CO2. It is a full reaction is irreversible because of the release of gas in the reaction. So here's 2 simple examples of it. So here's our ketone. So this is the alpha position, that's the beta position. Simply allowing this to come up to room temperature, and it spontaneously decarboxylates. Not only do beta keto compounds decarboxylate, but beta diacids. So we have a carbonyl here, it just happens to be a carboxylic acid. We have another carboxylic acid over here. Allowing this to come to room temperature spontaneously decarboxylates. In this case, we end up with the carboxylic acid. All right, so how does this reaction occur? And this one is remarkably simple as to the mechanism of the reaction. So here's what happens. If we draw the carboxylic acid in this form, we get a concerted rearrangement of electrons to give rise to what was referred to as an enol and the loss of CO2. So the decarboxylation step is a concerted one-step reaction that involves that rearrangement of electrons. Now, the immediate product is not carboxylic acid. The immediate product is the enol form. And so typically you need a little bit of acid to catalyze the conversion of this from the enol form into the keto form of the carboxylic acid. That happens by protonation on carbon at the alpha carbon. Produce this structure, and then deprotonation to reveal the carbonyl. And that's decarboxylation, in this case, of a beta diacid. One can draw the mechanism, and I'll leave it up to you, for the decarboxylation of the beta keto acid. And so it happens in much the same way. So finally, I want to leave you in this chapter with a little look at some carboxylic acid chemistry that's of physiological interest, and specifically it's this this final reaction that we just talked about, this decarboxylation reaction. It has some interesting physiological consequences. So in the grand scheme of things, the human body uses three basic metabolic fuels, and we're going to consider two of them very closely. The three basic fuels are carbohydrates, which are stored in our bodies as a polymer of glucose called glycogen. So this little cloud here represents the glucose that we store in our bodies in some form. This is largely in our liver, but a smaller extent in the muscles and surrounding tissues. Our second primary fuel are fats, or fatty acids more specifically. And we store those as what are called triacylglycerols. Those are that structure of fats that I showed you at the introductory slide in the chapter. To a much smaller extent, we utilize amino acids as fuel, but primarily amino acids are metabolized into proteins and those sort of things rather than burned for energy. So we're not going to consider those here. So what I want to talk about is an interesting phenomenon, and the interesting phenomenon is that the smell of acetone can be discerned on the breath of individuals suffering from extreme starvation or untreated type 1 diabetes. And the question is, where does the acetone come from? And it really is acetone. That's the reason that it's smelled. So if we think about the processing of these fuels and kind of on a day-to-day basis, the triacylglycerols that we store in our tissues in our body are broken down into fatty acids to be used as fuel. Glycogen is broken down into individual glucose molecules to be used as fuel, but primarily both of these things feed into the same metabolic pathway. They become a common intermediate in what's known as the citric acid cycle, and the citric acid cycle is the engine that drives our bodies. Very importantly, to fuel the brain, our body almost exclusively uses glucose, because glucose can be shuttled across the blood-brain barrier, but fatty acids and triacylglycerols cannot. We think of the brain as being metabolically segregated from the rest of the body. It's a defense mechanism. And so it can only use fuels like glucose that can pass the blood-brain barrier. In the case of starvation, the first thing that we use up is the thing that's most easily metabolized. So if we stop eating for extended periods of time, we will exhaust our stored glucose, and we get to a point where the body, in order to fuel the brain, then needs to make glucose. And the way that it makes glucose is to take stored fat, which it keeps in reserve for times when the glucose is used up. It takes the stored fat and it shuttles the fat into fatty acids, makes acetyl-CoA, and instead of shuttling the acetyl-CoA off into the citric acid cycle, it uses it to synthesize glucose in order to get fuel across the blood-brain barrier. This process, going from acetyl-CoA to glucose, is called gluconeogenesis, meaning making glucose. And it happens primarily in the liver. The problem is that it is energetically costly. It takes a lot of energy for the body to do this and synthesize the glucose in order to just get it through the blood-brain barrier. And so this process is supplemented. And the way that it's supplemented is to make less energetically costly fuels that can get across the blood-brain barrier. And these fuels, which are only made by the body in times of distress, typically during extreme starvation, these fuels have structures like you see here, and they're generically called ketone bodies because they have in them a ketone, for instance. The immediate, the first thing that's synthesized that is actually metabolically useful is this beta-keto carboxylate. And this can also be reduced to this alcohol. This can also be metabolized by the brain, but that's even more costly because that's additional steps. These molecules are small enough and readily diffuse across the blood-brain barrier that they can go into the brain, and the brain can metabolize those and get some energy out of those. The problem is this. If you look at that structure right there, that's what we've just been talking about. It's a thermally unstable molecule. So if it gets protonated, then, not surprisingly, it will decarboxylate. And this does happen. So in times of extreme starvation, your body makes this stuff in order to get it to the brain, but some of it spontaneously decomposes, produces acetone. We can't metabolize acetone directly. What happens is the blood circulates through the lungs. The vapor pressure of acetone is high enough that it simply diffuses into our lung cavity and we exhale it. And so you smell it on the breath. People who are suffering from extreme starvation. It's in fact one of the signs. It's a symptom of exhausting essentially your fuel sources, and you're down to very little to metabolize. So what about type 1 diabetes? Why do you observe the same thing? Well, type 1 diabetes is a curious phenomenon. It's an autoimmune disorder where the body's own immune system actually is destroying insulin-producing beta cells in the pancreas. Insulin is a hormone that is released to trigger the body to increase the amount of glucose. So essentially, when your body releases insulin, it does so to say, oh, we need to store the glucose is glycogen. So the insulin basically is driving this pathway, pushing the glucose to be stored. Type 1 diabetics don't produce enough insulin. And because they don't produce enough insulin, the signal isn't sent to take the glucose and store it. What happens is that the glucose builds up, and so you start producing too much. So insulin stimulates the synthesis of glycogen, and to some extent simulates the synthesis of triacylglycerols, and it inhibits their breakdown. So it tells our body, store these things away. We need to save them for a rainy day. So your body releases insulin, for instance, after a meal. If you have low insulin levels, that signal doesn't get sent, and so what happens is the reverse pathway tends to run kind of uninhibited. And so with low insulin levels, the breakdown of glycogen and triacylglycerols leads to an excess of acetyl-CoA. So your body kind of artificially produces too much of the fuel that needs to go into the citric acid cycle. So then the question is how to get rid of it. And what your body does is the same thing it does in times of starvation. It starts to shuttle it into other pathways because it's got so much of it. Now, a lot of that leads to an increase in glucose. So one of the symptoms of untreated type 1 diabetes is you have high glucose levels in your blood, and so the finger prick test to test for glucose levels. The other symptom of untreated type 1 diabetes is the smell of acetone in your breath, because this stuff builds up. You're making ketone bodies to try to get rid of it, not because your brain needs it, you got plenty of glucose, but you're just trying to get rid of this stuff because it's building up. And so if you make some of this beta keto acid, it's going to decarboxylate and you'll smell acetone on your breath. And that's it. That's the end of our chapter on carboxylic acids, and here's the usual list of study problems.